Hello and welcome to the next A-level lesson from the conscientious biologist Ben Gallagher. This is the first one in a really important sequence of lessons on proteins. This one looks at protein biochemistry, which basically breaks down to the structure of amino acids, how amino acids can polymerize to each other so that they can make chains, which then go on to fold into the proteins that we know and which we've studied at GCSE. This one covers those structures and it covers protein folding. This one isn't about protein formation and protein synthesis. That comes next. As always, please do subscribe to my YouTube channel if you haven't done so already, and do have a look at all the information that I put on Facebook and Instagram. Thank you. Right, we're going to do a really quick whiz through of two slides that are just basic GCSE stuff, but it's stuff that you must know if you're going to understand the rest of this lesson and what's in this sequence of lessons. So let's think about cells. What are they? What do they need to do? And we'll keep it really simple that they're very small compartments that can make specific proteins which do everything the cell needs. That's basically all we're talking about with a cell. It's a small compartment that produces proteins because the proteins will carry out metabolism. So to have a cell, you need two structural features. We've got the membrane and we've got the cytoplasm. The membrane's the skin, it holds it together. It controls some things entering and leaving, which we should have seen in the membranes lessons, which you should have already watched. And you've got the cytoplasm, which is just a liquid. It fills the cell to allow everything to float about and interact so you can get the chemical reactions and interactions that you need. The two things then to make the cell metabolically active will be the DNA or chromosomes of DNA, because chromosomes are the structures. They're the instructions for how to make proteins. And you need something that can follow those instructions and actually build them. Those are the ribosomes. Now, you'll remember from the cell biology lessons that you should have looked at in my A-level series. It's a lot more detailed than this in a cell, but these are the absolute key components. These four things alone will allow a cell to be metabolically active and show life, okay? Because it can make proteins, and as I said so often in my GCSE lessons, proteins do everything. So let's do a really quick recap of protein synthesis so we can put the rest of this lesson in context. So if you've got a ribosome, and this is directly from my GCSE lesson, so it's really straightforward. The ribosome will read the genetic instructions. Those genetic instructions just tell it a sequence of amino acids to put together in a strand. Once you've got those amino acids in a strand, that's called a polypeptide. The polypeptide will then, so it'll be very long, much longer than I'm showing here. If this blue line's representing the polypeptide, it will then fold up in a very specific way, and how it folds up will determine its shape. So the really important bits I'll summarize in this box for you, okay? The sequence of DNA nucleotides determines the sequence of amino acids in the polypeptide. You should have already watched my DNA and nucleotide biochemistry lesson, so that should be familiar to talk about nucleotides. But it's that sequence of nucleotides will determine the sequence of amino acids. The sequence of amino acids determines how that polypeptide will fold. How it folds will determine its shape, and its shape determines its function. That's true of everything in life. I could pick up any object around me. I could even look at my own hands, ears, nose, anything. It's their shape that allows them to do their job so well. So really, we're breaking this down to the cells have to be able to produce proteins. For those proteins to do their jobs, they need to be the right shape. To be the right shape, they need to be the right amino acid sequence. To get the right amino acid sequence, you need the code in the DNA. That's your absolute minimal basic understanding that you should have, even from foundation level GCSE. So now we're going to start upping the detail to A-level standard. So we've just seen that amino acids are the basic fundamental building block for how you make proteins. The ribosome reads the DNA and goes, right, I need that amino acid first, so I'll grab it, I'll put it there. So we need to know what their structure is. So let's just put one bit of information up. This is from the lipids lesson, which was in the membranes lesson that you should have watched. It's just some key bits of biochemistry to remember. So carbon atoms always have 4 bonds coming off them. Oxygen always has 2, hydrogen always has 1. Now that's what I gave you for the lipids, because lipids only contain those things. There's one extra element that I'm going to pop into that gap that you have in amino acids, and that's nitrogen. Nitrogen always has 3 bonds. So if you look at how I've set that out above me, starting from the one closest to me, it's 1 bond, 2 bond, 3 bonds, 4 bonds. They go up numerically. If you look at the first letter of the element, we've got HONC, it's honk. Okay, I know it's a C and not a K, but if you can remember honk, and you remember honk, honk, hydrogen, oxygen, nitrogen, carbon, 1 bond, 2 bond, 3 bond, 4 bond. Then anytime you're drawing a biochemical, you can just think, does it agree with honk? Check all your elements, check they all fit. That'll tell you if you've drawn it correctly. Starting off with an amino acid though, we've got a carbon right in the center. Now to the side of that, so the side closest to me here, we've got this group. I'm hoping you recognize that group because you've seen it before. That was the group that was on top of a fatty acid molecule that you got in lipids. It's the carboxylic acid group. If you look at the structure, yes, I've flipped it on its side for my amino acid, but it's the carbon double bonded to an oxygen with an OH group of it. So that bond to the side of that carbon in my amino acid, that's a carboxylic acid group. Over to the far side of that middle carbon, I've got a second group. I wouldn't expect you to know this one, but just look, it's got the nitrogen in the middle. We know nitrogen needs to have 3 bonds. It's got 2 bonds to hydrogen. It's got 1 back to the carbon. That group over there is called the amine group. So we've got the amine group over the far side, carboxylic acid group on this side, amino acid. That's why they're called amino acids, because they've got an amine group and a carboxylic acid group. Now look at the numbers that we need up here. Look at the middle carbon over there. We can see the middle carbon's got 2 bonds going out, so it's got 2 bonds going spare. One of those bonds, and it doesn't really matter whether you do it above or below, but one of those bonds goes to a hydrogen. The second bond goes to this. This is called the R group. Okay. Now everything that I've drawn on there so far, every single amino acid has, but the R group can be different on different amino acids. And if you think back to the previous slide we looked at, where I used those little emoji smiley faces that I used at GCSE and in previous lessons to represent amino acids, the R group is kinda that smiley face. It's the face of the amino acid that makes it different. So if I run through the different faces, like you can see, it's just that R group that can be any one of those faces. That's what makes it different. That's what makes it different. That's why it's called a variable group. Okay, now there's hundreds of different variable groups that exist. However, there's only about 20 that we need to know about. So whilst there are hundreds of different types of amino acids, there's only 20 amino acids that are coded for by our genetic code that we use and we incorporate into our proteins. So take a screenshot of this, but you must be able to draw out an amino acid structure just like up there. Let's look now at how those amino acids can join together, and they join together by something called peptide formation, because they make a peptide bond between 2 amino acids. So if I pull one amino acid up over there, it's exactly the one that we've just been talking about. If I put another one directly next to it, you can see that I've got the OH of the carboxylic acid group of the far one right next to one of the hydrogens on the amine group of the second amino acid. So the bond formation is going to happen within that yellow circle. Now look at what I've got in that circle. I've got 2 hydrogens and 1 oxygen because of the OH and the H. 2 hydrogens, 1 oxygen, H2O. It's exactly like we saw in the triglyceride where we formed the ester bond, where we've got an H2O to give away. If you lose an H2O from a bond, then it's a condensation reaction. We talked about that with the ester bond. The difference here, though, is in the ester bond, it was 2 OH groups. So when you lost H2O, there was still an oxygen left over. So that oxygen made a bridge between the fatty acid and the glycerol, gave you an ester bond. Here, because we've only got 1 OH group and then a hydrogen, we don't have that oxygen left over. You've just got 2 exposed bonds there. The carbon over there has got a bond spare, the nitrogen here has got a bond spare, so they just bond directly to each other. That brown link there, that's your peptide bond. And when you're describing it, you ought to be using as much detail as you can. I say that so often in my videos. Get detail into your explanations. So if it says in the exam, describe a peptide bond or explain how a peptide bond forms, I want something like this. Okay, a peptide bond forms by a condensation reaction between the carbon of the carboxylic acid group on one amino acid and the nitrogen of the amine group on an adjacent amino acid. So we're saying exactly what it forms between, how it forms. Key bonus point, water's given off in this reaction. Always worth a mark in the exam. 5 specific points there. Please don't just write in the exam, explain how a peptide bond forms. Oh, it's when 2 amino acids join together. It's going to be worth 0, 1 at most, okay? Use detail when you're giving your answers. Now, on the diagram here, obviously, the amino acids are quite far apart. I've made the bond very long. It's not really that long. If we look at this one, this is more representative. You can still see the brown peptide bond in the middle there, holding 2 amino acids together. When there's 2 amino acids, it's called a dipeptide. The di bit referring to 2, peptide referring to them being peptide bonds. Please note though, a dipeptide contains 2 amino acids. It doesn't contain 2 peptide bonds. So dipeptide is a little bit misleading. You look at it there, it's a dipeptide, 2 amino acids, but it's 1 peptide bond. Okay. Now, if I extend this one, if I get rid of the OH that's there and I add on a couple more amino acids to the side, I've now got a line of 4 amino acids. Just check that you can identify the 4 amino acids. Look at where the peptide bonds are. Now, really importantly, and I say this so often to students when I'm teaching this, it's 4 amino acids long. So I say, how many peptide bonds does it create? And they almost always go, well, 4. But it's not. Remember, the first one didn't have a peptide bond. You peptide bonded the second one to it, then the third, then the fourth. So there's always 1 less peptide bond than there are amino acids, which means when this tetrapeptide, because there's 4, when this formed, it gave off 3 molecules of water. Okay, so in the formation of this tetrapeptide, 3 molecules of water were formed, 1 from each peptide bond. Or to put it another way, if you wanted to break this very short polypeptide, because it's only a tetrapeptide, but if you wanted to break it down, so to do hydrolysis, which is the opposite of condensation, you'd need to add 3 water molecules. So just be aware, it might say in the exam, there was a polypeptide made of 192 amino acids. How many peptide bonds would it contain? Well, 191, because there's always 1 less. How many water molecules would be formed? 191, because it's 1 less than the total number of amino acids. Hopefully that's obvious, but don't fall into that trap in the exam. Okay, what we'll do now, we'll start to look at how these really long ones can start to fold up to give you the proper shapes of your finished proteins. So let's start off with a really long polypeptide going on there. I say really long, it's only sort of 10 or 12 amino acids long, but much longer than the ones we've been looking at. But if I've got my amino acid chain there, okay, my polypeptide, poly meaning many, peptide bond, many bonds, okay, it's still, by polypeptide standards, very, very short. So if I was going to talk about this a little bit more realistically, then that entire thing would only be at the very start of a fairly standard length polypeptide, and we call that its primary structure. So when it's just in the straight line polypeptide, that's the primary structure. It's just the polypeptide strand. The amino acid bonds are held together by peptide bonds. That's it. That's all that's going on. To start it folding up, to start to achieve its shape, it needs to go from its primary structure to a secondary structure, which is loose folding held together by hydrogen bonds. Now, hydrogen bonds are attractions between positively and negatively charged parts of molecules. Now, if you look at the amino acid strand up there, look along the top next to the R groups, you've got the double bonded oxygens and the single bonded hydrogens. Now, the double bonded oxygens at the top there, because they're quite large, they pull electrons towards them. So the oxygens that are sticking up from the carboxylic acid groups, they've got a bit of a negative charge. The hydrogens, because hydrogens are very small, their electrons get pulled towards the nitrogen. So they are slightly positive because electrons are negative. So the oxygens, in having more negative electrons, become a bit negative. The hydrogens become a bit positive. So all along a polypeptide, you've got alternating positives and negatives. So you could just take it and just fold it over and then find positive and negative attractions and just start forming links. Now that allows it to loosely fold up like the structure I've shown you over there. But if you look at that, you can, or you should, be able to identify certain structures within it. If you look on the furthest side away from me on that secondary structure, you can see it coils round and round. That's called an alpha helix. That's a fairly typical pattern to see repeating within a secondary structure of a protein folding. It just forms kind of spirals. Next to that, a little bit closer to me, you can see it goes up and down and just pleats itself. Well, those are called beta pleated sheets. Again, very common to see in secondary structures. Above those, you can see that it's just more random. You get random folding that just happens in secondary structures as well. The point of the secondary structure, though, is it pulls everything a bit closer together. The R groups are then all closer together so that you can move to the next stage of folding, which is to get a tertiary structure. This is the important one. This is the complex and specific folding, and this is directed by the R groups, because some R groups want to be next to and bond with certain other R groups. So if you think about all the R groups that I've got running along the top there, think about when I had them as emojis, maybe the ones with the moustaches, they want to be together. So if there was one with a moustache over this end and one with a moustache over that end, then they might fold so that they can get together and bond. So what we're talking about is because the R groups want to be with certain R groups, they'll bend and fold the whole thing to get next to the one they want to be next to. And when they do, those two R groups will form either ionic bonds, as I've written here, or they might form something called a disulfide bridge, both quite strong bonds that will do the fine folding and give you your finished structure. So that one over there, that little highly folded brown blob now, might be the finished folded enzyme or whatever it's going to be. Now there is one more bit that we need to add to this, because whilst we've got on here primary, secondary and tertiary folding, there is something called quaternary structure. Okay, now quaternary structure, even though the words primary, secondary, tertiary, quaternary come one after another, it's kind of a little bit wrongly named, because quaternary doesn't really come after tertiary. Quaternary is something a bit different. It comes from secondary. It's another option from a secondary structure, because a quaternary structure is the same as a tertiary, but it contains either more than one polypeptide or a prosthetic group. A prosthetic group is just something extra, something that isn't an amino acid, okay, a different molecule from somewhere else. So if I put at the top here, there's my original brown polypeptide, there's now a blue polypeptide with it, and that kind of green star is representing just a prosthetic group, something else that isn't an amino acid, okay. Now when they fold up into their secondary structure, they do it together, and then when that folds up into its, what would have been its tertiary, they all kind of join together. But because there's more than one polypeptide and a prosthetic group in this, we call it a quaternary. So what I'm basically saying is you can go primary, secondary, tertiary, or you can go primary, secondary, quaternary. What you don't do is primary, secondary, tertiary, quaternary. It's primary, secondary, and then depending on how many polypeptides or whether it's prosthetic groups, that determines whether it moves on to a tertiary or a quaternary. Hopefully that makes sense. You certainly need to know the sequence of names. You need to know what bonds are involved in those structures, and you do need to know the difference between a tertiary and a quaternary protein. Let's look at a final slide now for a little bit more nomenclature, a few more names that you need to know about proteins. We can be pretty quick because it's only two names: globular or fibrous. They are the two main types of proteins that you can get. So whether it's a tertiary or quaternary structure, the purpose of that finished protein will determine whether it's globular or fibrous. So globular proteins are metabolically active proteins. In other words, they react with things, okay? For example, enzymes. Enzymes are globular proteins. So amylase, helicase, DNA polymerase, any of the enzymes that you've ever learnt about, they're all globular proteins because they react with a substrate, they create products, they're metabolic. Now, key hint for the exams, if it asks you for an example of a globular protein, don't write enzymes, because enzymes is a massive group, it's not an example. If it asks you for an example, put the name of an enzyme, put amylase or helicase or whatever. Don't just put enzymes, okay? Another good example of globular proteins, or an example, is hemoglobin. So hemoglobin you've studied, it's what carries oxygen around the body. It can bind to oxygen atoms. So because it reacts with oxygen, it reacts with something and can unreact and give it out, switching from hemoglobin to oxyhemoglobin and back, it's a globular metabolic protein. So this is what hemoglobin looks like, or is a model of it, with the different chains there. So you should be able to see it's a red chain, a pink chain, a purple chain, and a sort of pale purple chain. And if you look very closely, you might be able to see kind of little red things floating about within it. Those are the prosthetic groups, which are iron atoms. That's what allows it to carry oxygen. So because it's got multiple polypeptides and prosthetic groups, we can say that hemoglobin is a globular protein for those reasons, but it's quaternary as well. So it's a globular quaternary protein composed of 4 polypeptide chains plus iron prosthetic groups. I suggest you learn that one because quite often in the exam it says, give an example of a globular protein. Sometimes it says, give an example of a quaternary protein. If you remember hemoglobin, you can tick both boxes because it's globular and it's quaternary. Okay, fibrous proteins, they're just structural, so they don't do metabolic jobs, they don't react with chemicals. You can build stuff out of them. Okay, so fibrous proteins are structural. For example, collagen. Here's a collagen molecule. What we've got there, you can see blue, pale green and light green. You've got 3 different polypeptide strands here, and they spiral round each other so that it makes kind of like a triple spring. It makes like bioelastic, it's stretchy. Collagen is the molecule you have in your skin that makes your skin kind of stretchy and ping back into place when you pull it. It is literally the elastic part of your skin. So it's a really important structural protein. And as we've said, collagen's a fibrous, quaternary because it's got those 3 polypeptides, quaternary protein composed of 3 polypeptides. So these are just good examples to use, more key terms of globular and fibrous to go with your primary, secondary, tertiary and quaternary, and your structures of amino acid and your polypeptide formation. So that was really essential groundwork to allow you now to go on to the really detailed processes of transcription and translation, which are how proteins are synthesized in the nucleus and then by the ribosome to get the polypeptide chains. And we've just seen how those form and how those fold up. So this is now the really important video to go to look at protein synthesis. Please do subscribe to my YouTube channel if you haven't done so already. Give this video a like if you found it useful, and please remember to go and have a look at my Instagram and Facebook pages. There's lots of extra information that can supplement your course and really useful for the synoptic paper when that comes up in year 13. Thank you.